Hey, hey, hey. All right, go ahead and get out your notes for Molecules of Life part 2. So go ahead and get those out. It's Molecules of Life part 2. Okay, so today we're talking about protein and DNA. So remember from yesterday's notes was Molecules of Life part 1. So yesterday we talked about carbs and lipids. Today we're talking about protein and nucleic acids. And that's the 4 molecules that are in all living things. There's carbs, lipids, which are fats. And then today we're going to learn about proteins and nucleic acids. So I want you to know that there's 4 molecules of life. 4 molecules or 4 macromolecules that are in all living things. So we'll start with proteins first. And it says a protein is also called a polypeptide. Poly means many. You should have filled that in right there. Poly means many. And proteins are made up of the bonds. The covalent bonds are called peptide bonds. So many peptide bonds is another word for protein. So polypeptide means many protein bonds. So polypeptide, many protein bonds. Okay, so proteins and enzymes. Enzymes are proteins, but an enzyme is a protein with a special job. A job like breaking a molecule down, something like that. So proteins make up greater than 50% of your dry weight. So if you took out the water, over half of you is protein. So all of us are made up of protein. We have lots and lots of proteins in us. It says this is an example of where the structure of something determines what it does, the function of it. And so proteins, they fold, and their folding has a lot to do with what that protein does. Okay, proteins are made up of amino acids. So if you see the word amino acid, highlight that word. Amino acids are monomers. Proteins are polymers. Okay, so another word to say that would be a bunch of amino acids make up a protein. So if you see a bunch of amino acids all balled together, folded up, that makes up a protein. It says individual amino acids are bonded together by peptide bonds. So peptide bonds is amino acid peptide bond, amino acid peptide bond, amino acid peptide bond. And so there's a protein all folded up. But amino acid and then peptide bond, that's where it gets the name polypeptide. So amino acids make up proteins. Amino acids are bonded together by peptide bonds. Okay, so if you look right here, this is an amino acid. So amino acids are made up of, and we're going to learn this in class tomorrow too. Amino acids are made up of this C and O group. You can see here. And then they're made up of this hydrogen and nitrogen little group over there. And then, do you see what's in the white? That's an R group. And that white box can be anything. It can be 20 different things. So you can see here they're all different. Okay, so you got the amine group. And amine group, that's the H3N. The carboxyl group is the C, the double bond to the O, and then the single bond to the O. And then these are hydrophobic. Remember what the word hydrophobic means. It means it's water fearing. So all of these are hydrophobic. Okay, well, there's more. And you don't have to memorize these structures or anything like that. Okay, here's some more. And these are all hydrophilic. You don't have to memorize those, but notice that everything in the box is white. I mean, in the box, that's white. That's the R group. That's the different thing. So the amino acids are just alike, all except for the R group. Okay, well, it says these are hydrophilic. They like water. Why? Okay, well, they have different charges. They all have different charges, and that's the answer. They have charges. Some are positive, some are negative, some are polar. So let's talk about right here where the word polar is. The word polar means it's a molecule that has a negative charge and a positive charge. So water is a polar molecule. So do me a favor and write that in. Water is polar. And then define polar. Polar is a molecule that has a positive side and a negative side. Okay, so of something that's positive and something that's negative inside one molecule. Okay, let's talk about now, how does a protein fold? We said it has to fold in order to work. It has to fold in order to work. And so this picture is just showing you there's an amino acid and there's an amino acid, and they're bonded together. What's the name of the bond called? That yellow mark, what's the name of the bond called? It is a peptide bond. That's the name of the bond. Okay, and this picture is showing you how water is removed. You should know this. Water is removed, puts molecules together. What's it called when water is removed and it puts molecules together? That's called dehydration. Should have learned that in class today, or from last night's video. Okay, so how does a protein fold? After a protein's made, it's got to fold. The primary sequence is just a straight line of amino acids. So it's like, if you look at this picture, amino acid, amino acid, amino acid, just a big old line. Amino acid, amino acid. You got peptide bonds, amino acid, amino acid, amino acid. So all those amino acids are bonded together. Okay, highlight, this has covalent bonds or peptide bonds. Do you see? It doesn't look like it says that. So beside primary structure, do me a favor and write in just peptide bonds. And this sequence is really important. The order of the protein is really important because that's what protein it is. It says it refers to the structure that it does. So A says this refers to the sequence of bonded amino acids. And you'll see where it says it's really important because that determines what the protein is. So remember from the first unit, DNA is what? DNA is the instructions to make proteins. Good. DNA is the instructions to make proteins. So the order of your DNA would determine what protein you made. So let's talk about, for example, you have a protein that you know of already is a microtubule. That's a protein in the cell that your DNA makes. So DNA is like the recipe. It makes the microtubules, and microtubules are what move around chromosomes inside the cell for mitosis. Well, if you don't have the DNA to make the appropriate microtubule, then you wouldn't be able to make that. So you wouldn't have the recipe. Your body doesn't have the recipe to do that. Well, the recipe tells you what order to do the steps in. Okay, well, so your instructions is what order to put the amino acids in. Okay, well, here's another example. Sickle cell is a genetic disorder where the hemoglobin molecule isn't made, or it isn't made correctly. And so someone who doesn't have sickle cell, their red blood cells look like this, nice and big and red. Well, someone who has sickle cell, their red blood cells are shaped like a crescent moon. Do y'all see how it's shaped differently? And it hurts when it goes through the arteries and veins. And all it is because one amino acid is wrong. See how it's supposed to be the abbreviation GLU. Instead, it's the abbreviation VAL. So the DNA, they have a miscode. They have one letter of their DNA is wrong. So they don't make the right protein. And you can see where it affects all the red blood cells. Okay, so primary structure, you want to know, it's a straight line of amino acids. It's amino acids stuck together with peptide bonds. Okay, secondary structure. There we go. Secondary structure is where it folds up and it says hydrogen bond. So that amino acid starts to fold up. It says between neighboring amino acids. They fold and they overlap. And do you see where it says coil? See how it looks like there's coils like Arby's curly fries. So the secondary structure has got some Arby's curly fries in it. No, not really. But it's got that straight line starts to fold up because of hydrogen bonds. The hydrogen, look right here, this is the first one. The hydrogen starts to coil. Okay, tertiary structure is the third structure, and it just a variety of bonds fold together. And really these bonds deal with sulfur. So those hydrogen bonds are on the secondary structure, but now the sulfurs start to bond together, and they're called disulfide bridges. So that's two sulfurs stuck together. Two sulfurs stuck together. And you see the word disulfide bridge. Okay, and then quaternary is where more than one polypeptide or more than one protein goes together. So a bunch of them. So this is where two or more polypeptides are woven together. So many proteins go together. Hemoglobin have 4 proteins together. So that's hemoglobin right there. And you can see you got this ugly yellow color and this ugly brown color. And then kind of this purple color or whatever. And then 4. So there's 4 of them all together. 4 make... It's just 2 or more, but in this case it's 4. So there's... Okay, let's review this right quick. Proteins have to fold 4 different steps in the folding. The primary one is first, and that's a straight line. Secondary, that's got the hydrogen bonds. Tertiary, that's got the, remember where it said disulfide bridges? And then the fourth structure is where more than two or more amino acid sequences go together. And sometimes if a protein gets too hot, it may unravel or unravel or unfold. See how this one is denatured on the right? So denature is just the unfolding of the protein. Unraveling is what the notes say. Denature can be caused by like an acid or salt or temperature. So something that's a strong acid may make it unfold. Okay, and it says the most common bonds under denaturing that have been affected during denaturing are the weak hydrogen bonds associated with secondary structure. Okay, so let's just review really quick hydrogen bonds. Let's just review those hydrogen bonds really quickly. Okay, so all about proteins. We're just going to review proteins. First thing, they're made up of amino acids. They've got polypeptide bonds. The way they fold is important because that determines their function. Okay, and then they fold. The primary is the first one. The secondary is where it look like an Arby's curly fry with hydrogen bonds. The tertiary is where it folds up because of sulfur. So it's really folded up. And then the fourth one is quaternary, and that's where 2 or more polypeptide chains go together. So that's what makes a big functioning protein. So primary, secondary, tertiary, quaternary. Okay, and remember that primary, the order of the primary sequence is important to determine the function. The folding is important to determine the function. If the protein were to unfold, it's called denature. Good. So hopefully you're good on proteins. Okay, we're now moving on to nucleic acids, which is going to be DNA and protein. So here's a picture of what DNA looks like. The monomer of DNA is called a nucleotide. Okay, so see that word in your notes. A nucleotide is a monomer of DNA. So what is a nucleotide? That's where we're at now. Okay, if you look at this picture, a nucleotide is made up of a phosphate, a 5-carbon sugar, and a nitrogen base. So go ahead and let's draw this in your notes. Like flip it over or draw this in your notes, and I'm gonna draw it again so we get it. Okay, you've got I'm going to draw it again. I'm going to label it. And you're going to draw it too. So go get you a pencil if you don't already have one. Okay, you've got a phosphate group, which is just like a phosphorus from like the periodic table of phosphorus with oxygens all around it. A 5-carbon sugar. Looks like a house. There's 5 sides. So 5 carbons. See, 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. So this is your sugar. And then a nitrogen base coming off this way. Okay, so that nucleotide you just drew, the phosphate, the 5-carbon sugar, and the nitrogen base, they make up DNA or RNA. So, back to this picture, you've seen on TV, DNA looks like a ladder that's twisted. And RNA, so that's what DNA looks like. RNA is like a ladder but only has one leg. So it's missing the other side. Well, those nucleotides make up the ladder. So look right here. It would go phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. So the phosphate and sugar are the backbone. The nitrogen base is the rung, like where you would put your foot if you were on a ladder. Okay, so these are sources of genes and heredity. These are sources of genes and heredity. So DNA is it says $1 million blueprint. But you and I know really what it is. DNA is the instructions to make protein. And remember when we did the unit on cells, we said that was the recipe for proteins. RNA is just a half copy. So go ahead and draw out the little DNA as the double-stranded ladder and RNA is the single-stranded ladder. RNA is cheaper. It's easy to make. It's disposable. RNA leaves the nucleus and goes out to the ribosome. Okay, so when you look, there's purines and pyrimidines. So E and F, purines and pyrimidines. C, cytosine, thymine, and uracil, uracil's on RNA, are all pyrimidines. Okay, and what a pyrimidine is this one little ring. See how C and T are one ring and A and G are 2 rings? Okay, well a purine, one ring, always pairs with a pyrimidine. Okay, so a purine has 2 rings. Purine has 2 rings. So purine always pairs with a pyrimidine. Okay, so let me show you how I remember this. Okay, so you're looking at the words purine and pyrimidine, right? Okay, well, if you look at the nitrogen bases words, cytosine and thymine both have a letter Y in it. And so does pyrimidine. So cytosine and thymine are pyrimidines. And that's a 1-ring carbon. It's got 1 ring, nitrogen base with 1 ring. Okay, the purines are guanine and adenine. See how those words don't have Y's? Okay, so purines, which are the 2 carbon rings, purines are 2, and they are purines, not pyrimidines. So guanine and adenine. Okay, so what does that matter? Well, a purine always pairs with a pyrimidine. So A always pairs with T. So A and T pair together, and cytosine and guanine always pair together. Okay, so that's in DNA. A pairs with T and C pairs with G. In RNA, there is no thymine. So think about it. In RNA, thymine is too big to come out of the nucleus. So in RNA, there's no thymine. So I scribbled it out. In RNA, adenine pairs with uracil. So uracil takes the place of thymine. So uracil is also a pyrimidine. Okay, so just to make sure you got this, uracil, thymine, and cytosine are all pyrimidines. Adenine and guanine are both purines. Okay, so look at the picture here, and you can see that okay. There we go. And you can see that a purine and a pyrimidine always pair together. Okay, so DNA, it says the sequence determines what protein or enzyme is made. And you really know that from the first unit. Okay, so DNA is a double helix, so you can see it looks like a ladder twisted. James Watson and Francis Crick came up with this. So they built the model. And model just helps us understand better, right? Help us explain concepts. So it says Watson and Crick built the model. And it says it's complementary. So it is A pairs with T, C pairs with G in DNA. So complementary. One side has information to make proteins and enzymes. One gene makes, a gene is a segment of DNA. One gene makes a protein or an enzyme. So it says the more nucleotide sequence genes in common, the more closely related organisms are. Okay, so take a look at this. This is a table just showing one protein, just one protein, not every protein, just the hemoglobin protein, just that one protein. The difference. So humans and humans, so it's a comparison of humans. So humans and humans have no difference. A gorilla and a human has one difference just in that one protein. Now we have lots of differences in our whole bodies. But in that one protein, there's one amino acid that's different. Okay? And you can see all the way down to a frog, there's 67. So we have more in common with a gorilla than we do a frog. So that's what this table is showing you. Alright, so I hope you got this. Let's review DNA really quick. DNA is made up of nucleotides. DNA is made up of nucleotides, and RNA is too. And DNA is double-stranded. RNA is single-stranded. And it's made up of these phosphate, a 5-carbon sugar, and then the nitrogen bases. The nitrogen bases are adenine, thymine, cytosine, guanine, and uracil. And it doesn't look like those words are in your notes. So if you want me to write them out for you one more time, I can. Adenine pairs with thymine. Cytosine pairs with guanine. Okay, and remember that the thymine and the cytosine are both pyrimidines. And adenine and guanine are both purines. And purine always pairs with a pyrimidine. Okay, so I hope this was helpful.